So this week we are going to look at a scenario that when you are finished with your studies, if you go into hospital administration or you open your own private practice, if you are a clinician, this is a very common scenario that you may encounter. In the exercise I gave you this week, Joseph Vasco from MedDev Incorporated is in your office on a Monday morning at 10 a.m., going on and on about a new device that his company came out with, the NeuroMesh stent system, and how it can significantly reduce the number of days for length of stay for stroke patients, as well as increase the number of days to a readmission encounter. He also goes on to mention that the NeuroMesh, once it is available on the market, will only add $7,250 to a patient's total charge. He goes on to make a special note of this because the NeuroMesh stent doesn't take any more time on the surgeon or the physician's behalf to insert, so there's no increase in physician utilization or surgeon utilization, extra supplies, time value units, anything like that. He goes on further to mention that the NeuroMesh can only be used in patients that fall into three very specific DRGs. DRG number 34, which is a carotid artery stent procedure with major complications or comorbidities. DRG number 35, carotid artery stent procedure with complications or comorbidities. Or DRG number 36, which is the carotid artery stent procedure without complications, comorbidities, or major complications or comorbidities. Vasco also provided you some information on the mean length of stay for each three of those DRGs, as well as a mean length of stay across all patients in the clinical trial across the three DRGs, which is 9.2 for DRG number 34, 5.8 for DRG number 35, and 3 days for DRG number 36, with a mean length of stay across all three DRGs and all the patients in the clinical trial as 6 days as their length of stay. Excuse me, and I'm sure as you can hear a little bit, I am sick. I do have a cold, so I do apologize if I have a coughing fit. I'll try and pause the recording so that way you don't have to listen to that. But if my voice does go in and out, I just want to apologize a little bit in advance for that. So continuing on the scenario that I gave you here, what would normally happen if you work in a large medical center? When device reps come in, or any kind of representative that comes in, they'll give you, data that you can go and identify patients in your EHR system and be able to do a statistical analysis to make a decision on whether or not you want to purchase MedDev's new NeuroMesh stent system or not, or whatever device rep happens to be in your office that day. So Vasco gave you the data that you gave down to your informaticists or your statisticians. They queried the EHR system and pulled out the cases that met the criteria. The ones that will be in the clinical trials, which will be identified as a variable in our data set, which we'll look at in a minute. As well as all of the other stroke patients that your hospital and organization has seen over a specific given amount of time. Your informaticist has given you a data set back to perform your own analysis because they know that you are a competent statistician and you can run some analysis on data. So as usual, the data set that you see in my recording here is a little bit different than the data set that you have. I have about 29,500 cases in my data set. And again, your data set has, more than 70,000, cases in your data set to run the analysis on. We do in this data set, excuse me, have two new variables to look at. We have this readmiss variable, which is the days, with day 0 being the discharge date for your patient, or, this patient number 1, this 80-year-old female patient was here for 4 days. And when they were discharged, they were out of the hospital for 119 days before they came back and were readmitted for another interaction. The information that we do not have on this readmiss variable is if they were readmitted, or when they were readmitted 119 days later, if they were readmitted for, another stroke intervention, if they were back on complications from their original stroke, or if it was something completely, totally different. So the readmiss variable, like I mentioned, is new in this data set. And then the second new variable, if you scroll over a little bit, she didn't have to, is mesh. So this is the variable indicator. It's kind of a twofold thing that you can consider in two ways, but in the questions I'm asking you, it really only has one meaning. The mesh variable will indicate, will have a value of 1 if the patient received the neuromesh. Let's see if I can find one. Yep, there is one right here. So we have a 57-year-old male patient who was in the hospital for 13 days and was readmitted to the hospital 149 days later. That did receive the neuromesh and participated in the neuromesh clinical trial. So like I mentioned, the mesh variable has two meanings, essentially. It means that they did receive the neuromesh and that they were participating in the neuromesh clinical trials. So let's start our analysis here. Excuse me. Let's start our analysis here by looking at the questions that I provided you. This one is a little bit longer than the other exercises we have done in the past couple weeks, but a lot of the questions you can use the same data for, run an analysis on question 2, and maybe you can answer 3 and 4 from the same output. Don't quote me on that. I just made that up. So question number 1. Which patient phenotype, phenotype 1 or phenotype 2, have the patients in the specified DRGs been coded under? So this means the phenotype 1 and phenotype 2 variables here, pheno 1 and pheno 2. This question is asking the patients that are in the DRGs that Vasco provided, so DRGs 34, 35, and 36, which phenotype variable is used to indicate that. So to figure that out, it is a little bit messy because we are looking at DRGs and there are 999 DRGs. So this one will take a little bit of scrolling when we get our output. But to do that, I would run a crosstab on that. Select crosstab. And then I would look at DRGs by phenotype. So I would put DRG in my rows, and column phenotype 1 and phenotype 2. Excuse me. So in this situation, I am going to actually add both of my phenotypes into the columns because all this is going to do is just give me two separate tables, DRG by phenotype 1 and then DRG is by phenotype 2. It will just be, like I mentioned, in two separate tables. All right, so let's go ahead and run this analysis here. And like I mentioned, there will be a little bit of scrolling. You can see how small it is here. But luckily, we know what DRGs we're looking for. So in patient phenotype 1, if we scroll down to DRG 34, 35, and 36, and in the 1 column, which means yes for phenotype 1, 34, 35, and 36 all show 0 patients. So if we then go down to the second DRG by phenotype table, DRGs by phenotype number 2, we'll look in the patient phenotype 2 column and then go down to 34, 35, and 36, DRG number. And we do see that our patients in my data set are coded in phenotype number 2. As always, like I mentioned, my data set only has about 29,500 cases in it for my analysis. So just like in the past few weeks, please do not use the answers that I'm giving you. They may or may not be different than the answers that you are going to get when you run the analysis on your data set. As I mentioned, your data set is more than 3 times bigger than mine, or about 3 times as big as mine is. So yes, please do not always follow the answers I'm giving you. Please run the analysis yourself. Okay, so that is question number 1, was which patient phenotype, 1 or 2, are my patients in. So I am concerned for the remainder of my analysis. When asked about phenotype, I am concerned with phenotype number 2. Question number 2, which of the DRGs identified in the phenotype from question 1 has the most patients? So for me, for phenotype 2 in my data set, the DRGs that has the most patients are DRG number 36. All right, question number 3. Is length of stay generally normally distributed? And when someone says generally normally distributed, they are looking for your entire data set. As you'll see a little bit later on, we will be filtering our data set to only look at a very specific subset of this, subset of the entire data set. But when someone says generally normally distributed, they are asking about the entire data set. So to answer this question, as we've done in weeks past, I would run a frequency table or frequency analysis on length of stay, and make sure in my statistics that I have mean, median, and mode, as well as skewness and kurtosis enabled. And in my charts, click the radio button for histogram and show me a normal curve. And click continue and OK. And we will get our frequency analysis. The skewness of my length of stay is 10.4, and my kurtosis is 2.28. I'm sorry, 228.49. So this tells me that my length of stay is certainly not normally distributed. I can confirm that by coming down to my histogram and seeing that this is not normally distributed. It's very rare that length of stay is normally distributed unless you're looking at a very specific ICD-9 or ICD-10 code that patients are in the hospital for a very certain amount of time for, and you need to look for outliers. Or if you're working for an organization that has a certain Medicare or Medicaid payment model that you're only given a very specific amount of reimbursement for a patient's interaction, which is supposed to continue across the whole continuum of care in treating that one specific interaction, or the diagnosis of that one specific interaction. But that information and doing those kinds of things are a little past what we need to worry about here. So please don't worry about that right now. So to answer question number 3, is length of stay generally normally distributed? My answer would be no in this case. Excuse me. Again, looking at the frequency statistics, the skewness and kurtosis, as well as the mean, median, and mode, they are not the same or close given their values. So question number 3, my length of stay is not normally distributed. Question number 4, what is the mean length of stay for each DRG identified in question number 2? All right, so for this one here, again, like we've done in weeks past, I'm going to do a compare means table. Set this, and I am going to look at length of stay by DRG, and let's run this analysis here. Again, this is another enormous table with upwards of 1,000 rows, but we are just worried about the mean length of stay for each DRG identified in question number 2. Again, those DRGs are 34, 35, and 36. So looking here, DRG 34, my average length of stay is 6.61 days. For DRG number 35, my mean length of stay is 4.32 days. And lastly, DRG number 36, my mean length of stay is 1.97 days. Right. So moving on to question number 5, what is the mean length of stay for patients in the phenotype identified in question number 1? So again, going back to question number 1, the phenotype of interest to me in my data set is phenotype number 2. So again, I'm going to run another compare means table and look at my length of stay, but this time I am going to use my patient phenotype 2 variable. Let's run that means analysis again. What is the mean length of stay for patients in the phenotype identified in question number 1? So patient phenotype number 2, I'm going to look at the 1 row because 1 means that my patient is in a phenotype number 2, and my mean length of stay for those patients are 3.25 days. Okay, moving on to question number 6. What are the mean total charges for the patients in the phenotype identified in question number 1? Again, same thing, we're going to run another compare means table. And this time I am concerned with not length of stay, but I'm going to use my total billed charges, or the TOTCHG variable. Put that up in my dependent list here. Click OK to run my analysis. And I see that patient phenotype 2 The total mean bill charges for patients in phenotype number 2 is $75,852. One thing I do want to note is that when you're reporting means of large currency values, unless you're in very specific industries or specificity is needed, you should always just round up or round down dependent on the decimal value, but only report the dollars. So the answer to my question in this case would be $75,852, not $75,851.91. So please just keep that in the back of your mind. That will be important later on. And this question number 6 is a two-part question. Is there a significant difference between mean charges of patients in the phenotype identified in question 1 and those who are not? So if you remember back to last week, we're going to need to look to find out first, to answer this question, if our total bill charges variable is normally distributed or not normally distributed. And as we've done that before in this recording today and in weeks past, I am going to run the frequency analysis on total bill charges, take out length of stay. I'm just going to make sure, again, I always just like to check to make sure that everything is still enabled that I'm looking for. Again, total bill charges, and click OK. And this one will often take a little bit of time. You have 29,500 cases in my data set, and this isn't a small value to need to calculate all of these statistics very quickly. So looking at my frequency table here, I can see that my mean, median, and mode are certainly not even close together. My skewness and kurtosis also aren't even close to 1. Let's go down to the histogram. And this, judging by the histogram, is also definitely not normally distributed. So now that I know that my total charges are not normally distributed, I am going to go ahead and run the same non-parametric test that I ran last week. So under non-parametric tests and independent samples, you will get this screen here. Just remember to click on fields. And then I am interested this time around in looking at total bill charges grouped by phenotype number 2. Because again, I'm interested in patients that are either in this phenotype or not in the phenotype identified in question number 1. So again, we'll click on settings, choose customize, and the Mann-Whitney U 2-sample test, and then we will click run. So output of our non-parametric test, our null hypothesis is that total bill charges is equal for patients that are in phenotype number 2 and patients that are not in phenotype number 2. And we can significantly, with significance, say to reject the null hypothesis. So we can say that the distribution of total bill charges is not the same across patients in phenotype 1 and that are either in phenotype 2 or not in phenotype 2. So again, just to recap, output of my Mann-Whitney U non-parametric test is telling me that with significance, P is equal to 0.000. The mean charges are significantly different between patients that are in phenotype number 2 and patients that are not in phenotype number 2. All right, moving on to question number 7. Question number 7, I gave you a little note just to mention about the mesh variable, and we're going to use that in the remainder of our analysis. So question number 7 would like to know how many patients received the neuromesh during clinical trials in your hospital. And to do that, we are just going to run a simple frequency analysis on the neuromesh variable. And that one, like I mentioned, is the variable is just mesh, but the description of the variable, if you hover over, is received neuromesh. So that is the variable that we're interested in. Just out of habit, I usually like to run the other statistics and stuff to go along with it whenever I run a frequency. A lot of the time that you don't need them, especially in this one because you only have 2 choices. But like I mentioned, they were enabled. Excuse me. They were enabled last time, so all right, we'll leave them. So to answer question number 7, how many patients receive neuromesh? The 1 value indicates that the patient did receive the neuromesh. So the answer to that question is 106 patients in my data set received the neuromesh or participated in the neuromesh clinical trials. Moving on to question number 8. All of the patients that received the neuromesh, were they selected from one DRG or were they selected from all of the DRGs that in my case made up phenotype number 2? So we're going to run a crosstab to figure that out. Let's do crosstab. And then we are going to reset this here. And again, I am interested in DRG. And for the column, I am interested in if my patient received the neuromesh or if they did not. Okay. And again, we do know that to receive the neuromesh, you have to be in DRG 34, 35, or 36. So we'll just scroll right down to those DRGs. And we can see that DRG 34 has 17, 35 has 33, and DRG number 36 has 56 patients. So to answer question number 8, no, patients that received the neuromesh were not all in the same DRG. Okay. Question number 9. What was the mean total charge for patients who received the neuromesh? To do that one, I would run a compare means table. And I would like to look at total bill charges, but in this case, I'm interested in patients who received the neuromesh and patients who did not receive the neuromesh. So put in neuromesh in your layer fields and click OK. So we can see here for all 29,442 patients in my data set, the patients that received the neuromesh, their mean total charge was $87,838, and patients that did not receive the neuromesh, their mean total charge was only $73,075. So this analysis right here should get you to start scratching your head a little bit. because in my meeting with Vasco, or in your meeting with Vasco, he told you that the NeuroMesh will only add $7,250 to the patient's total bill at the end. And as we can see here, there is a difference of about roughly $14,000. So, like I mentioned, this should get you to start scratching your head to wonder why these two means are so incredibly far off from each other. All right. Question number 10. Is there a significant difference in the total charge from patients in the same phenotype that did and did not receive the NeuroMesh? So for question number 10, this is the new topic that I am introducing this week. And it is filtering data sets within SPSS. We are interested in this time of total charges, mean total charges from patients in the same phenotype that did and did not receive NeuroMesh. So you kind of have to parse out this question a little bit to understand what it's asking. We don't have a variable that will let us do all of this in one step. So that is why we need to filter our data set to make sure that we're only going to get patients in phenotype number 2 to then be able to run our analysis from that point. So to build a filter, you go up to your data menu, then go all the way down to the bottom and choose the select cases option. I'm going to reset this real quick so it will look like yours. And then you get your list of variables here on the left, a couple of your filter options on how you want to, filter out your data set, as well as an output option on what you would like to do. So in our situation here, I am interested in patients that are only in phenotype number 2. So let me go back here. So I'm going to choose if condition is satisfied, and choose the if button, and then I'm brought to this window here. So this window here is called a filter builder, and what it lets you do is it lets you build a string that has to yield either a true or false result, or a 0 or a 1 result. That will allow SPSS to filter out the cases that you don't want to run an analysis on. So as I mentioned, we're interested in patients that are in phenotype number 2. So I'll go in my list of variables and choose patient phenotype 2. And choose this to put it in my filter builder. And then we already know that the variable has either a 1 or a 0 for patients that are in phenotype number 2. They will have a phenotype 2 value equal to 1. So what I would do here is when I add my variable, I will choose equals and then 1. And it is as simple as that. And choose continue. So now what I'm asking SPSS to do is select the cases if a condition is satisfied of phenotype 2 has a value of 1. And I would like you to just filter out unselected cases. So these 3 options here, filter out unselected cases. In my situation here, all 29,000 so odd cases in my data set would remain in my data set, and only the number of patients or number of cases that were identified as phenotype number 2 would remain as valid cases, and all of the cases that are not in phenotype number 2 are marked as invalid and are not processed. The second option here, copy cases to a new data set. What this option would do is any case that SPSS identifies as being true to our filter up here, it will copy out all of the variable data as well as all the variables themselves and build an entirely new data file for you to analyze. And lastly, delete unselected cases. This is something that I have very rarely ever used. It is a little dangerous because what this option will do is any case that does not meet your filter condition of phenotype 2 = 1 will be removed from your active data set. And once those cases are removed, it's often very difficult to get them back unless you have an alternate copy of your data set that you can just roll right back to. So to recap the filter or select cases window, I built a phenotype 2 = 1 as my filter condition, and then I would like to just simply filter out unselected cases, leaving my entire data set intact, but just marking certain cases as valid and other cases as invalid. So we will choose OK to filter it out. And we see our log here. And what we can do is go to our data window. And here we see that all of these cases have a slash mark through them. Cases that have a slash mark in them are not in phenotype number 2, which we can see that as valid here. And a patient that is in phenotype number 2, this patient right here, patient number 45. This patient does not have a slash through, which means they are included in future analyses until we turn our filter off. If you do use the filter option, one thing to keep in mind is that as you're running your analysis and then you're move on to a question that no longer needs the filter, one thing to keep in mind is as you're reading your questions, you have to pay attention that once you build a filter that you turn it off when you're done. It doesn't turn off automatically after each analysis is run. You are required to go in there and actually turn it off yourself. So one way to know that is, again, these slash marks here. If you were in your variable view and you were doing a quick analysis of the age variable, how can you tell from here that I have a filter enabled? And then to answer that question, it would be right down here. This filter on, I can't highlight it or anything, but down in the bottom right corner, it tells me that I have a filter on. So I would just need to be careful when I run anything not from the data view to have, just to make sure that if I've used a filter that I turn it off. And this filter on is also visible, it's just visible on that screen there. So when you're in the output, like I mentioned, if you start running some analyses and you notice that, an analysis that you've run before looks a little bit different or you're getting completely different answers, always just make sure that the question that you're trying to answer requires a filter, and if it doesn't and your answers are a little askew, just make sure that your filter is off if you don't need it. Right. So back to the question number 10 here. Is there a significant difference in total charges from patients in the same phenotype that did and did not receive the neuromesh? As we saw before, the total bill charge variable is not normally distributed, but just to be safe, I'm going to run that frequency table again for total bill charges with my filter enabled, just to make sure that when I filter down to a phenotype 2 and just include that those patients, that the total charges don't become normally distributed across patients in one phenotype. And then running my analysis, I can see my mean, median, and mode are not even remotely close. Same thing with skewness and kurtosis. These are not close or equal to 0. So I cannot, I can say that my total charge variable, again, at the phenotype 2 level, is not normally distributed. So saying that, again, I'm going to run another Mann-Whitney U non-parametric test for the total charges and patients that receive the neuromesh. So we will go down to non-parametric tests and independent samples. Reset this again. So my test field that I am interested in is total charges. And my grouping variable this time is going to be if I receive the neuromesh or if I didn't. So if we look back in our output window, back to question number 8, asking if all of the patients that received neuromesh were in one DRG or weren't in one DRG, we saw that there are patients across all 3 DRGs that did receive the neuromesh, and there are patients that did not. So the question number 10 that we're trying to answer now is looking at a different dimension of total charges, in that we're only looking at patients in phenotype number 2 that receive the neuromesh. So that is why we had to filter our data down to just including patients in phenotype number 2, because I can only group a this non-parametric test by one grouping variable. If I needed to do more, that would be an ANOVA table, and that's not something that we will get to. But again, just to recap, I am looking at my patient population now is just phenotype number 2 patients. My grouping variable is going to be if they receive the neuromesh or if they did not. And I'm looking to see if there's a significant difference between patients that received the neuromesh and patients that did not in total bill charges. So from here, again, let's just turn on the Mann-Whitney U test, because that is the one that I am interested in. We will let that run, and we get our output. So our null hypothesis of this test is that total billed charges are equal between patients that received the neuromesh and patients that did not receive the neuromesh within phenotype number 2. Our p-value for this is 0.002, which is less than our alpha value of 0.05. So that means that we reject our null hypothesis there, meaning that there is a significant difference in total charges of phenotype number 2 patients that receive the neuromesh and patients that did not receive the neuromesh. So I understand, and I get it, that I just gave you a lot of information all at once on filtering and why filters matter, and to make sure that filters are off when you're done with them, on and on. So I would recommend just watching the last few minutes of this recording again, just to make sure that you completely understand why we use filters and their purpose, and why I needed to run a filter before I ran this non-parametric test. Okay, moving on to question number 11. What is the difference in mean total charges for patients who received the neuromesh and those who didn't within the identified phenotype? So parsing this question apart, again, I do need a filter, and I happen to have that filter already enabled because I am asking for patients within the identified phenotype. So here, I want to know the difference in mean total charge of patients who received the neuromesh and for those who didn't receive the neuromesh, with my patient population being just those within phenotype number 2. And we have, we've done this before. We're going to run a compare means table. We'll reset it real quick. My filter is still enabled. I can tell that by looking here in my variable or my data view. My filter is still on. So my dependent variable is going to be total charges here, and my layering or grouping variable in this situation is going to be patients who received my neuromesh. We'll click OK. One way to, again, confirm that your filter is enabled is you should know that you have about 75,000 patients or so in your data set. I know that I have about 29,000 patients in mine. But when I ran this compare means analysis, my N is equal to 397 patients. So that should give it away fairly quickly that I do have a filter enabled on my data for analysis. So in this situation here, the total mean charges within phenotype number 2 for patients who receive the neuromesh, $87,839, and patients in phenotype number 2 that did not receive the neuromesh, their mean total charge was $71,486. And all this question is just looking for the difference between these two values here. Okay, moving on, question number 12. What is the mean length of stay for patients who received the neuromesh and those who didn't within the same phenotype? So just like the last few questions, when I parsed apart that question, I realized again that I still need my filter enabled to answer this question. So I need to again run another compare means table. Compare means. And this time I'm interested in length of stay in my dependent list. And I'm going to go ahead and take out the total billed charges. And again, I'm interested in the patients who received the neuromesh as my layering or grouping variable again. So we'll go ahead and click OK. And this is very interesting, actually. Again, length of stay by patients who received the neuromesh. Again, 397 is my N value, so I do know that my filter is still enabled for patients only in phenotype number 2. Going down to my output here. Average or mean length of stay for patients who did not receive the neuromesh is only 3 days, whereas patients who did receive the neuromesh in phenotype number 2, their mean length of stay was 4 days. Again, this should be another head-scratching moment, wondering why Vasco told you that his neuromesh product would significantly reduce the patient's length of stay, stroke patient's length of stay within your facility, when you're clearly looking here that patients in the phenotype that were able to receive this clinical trial device, the neuromesh, their mean length of stay was actually a day longer than patients within the same phenotype that did not receive the neuromesh. So again, something to keep in mind, something just to think about while you're running your analysis. Question number 13, is mean length of stay for neuromesh patients greater or less than patients in the same phenotype? We just answered that question. Patients that receive the neuromesh, their length of stay on average was 1 day longer than patients that did not receive the neuromesh within the same phenotype. The next question, question number 14, is the mean length of stay for neuromesh patients greater or less than all stroke patients within your data set? So this is the question that is asking, all stroke patients. So in this situation, I do need to go ahead and remove my filter, just so that way my next analysis that I run includes every patient. And I do that by going back to data, and then going down to the select cases option, and changing the radio button from if condition is satisfied up to all cases. So what will happen when I click OK with the all cases option enabled, it's going to go ahead and remove my filter, filter off. And I can confirm that by looking at my variable view. Down the bottom here, it does not say filter on. If I go over to my data view, none of my cases in my data set are struck out. So question number 14, is the mean length of stay for neuromesh patients greater or less than all stroke patients in your entire data set? So again, we're going to run the compare means table. Oops, sorry. Means. And I don't need to make any changes because I'm being asked for the same, dependent and independent variables here. Length of stay and receive the neuromesh. So I'll leave everything as is and click OK. And we can see, that I can confirm that my filter is disabled because now I am analyzing all 29,000 patients in this analysis that I run. And going down to my output here, I can see that my mean length of stay for patients that did not receive the neuromesh is on average 6 days, whereas patients that receive the neuromesh, in comparison to all of my stroke patients, their mean length of stay is only 4 days. So this again should get your head scratching a little bit. Why do my neuromesh patients that participate in that clinical trial, when looking at all of my stroke patients, why is the length of stay less than patients that did not receive the neuromesh? But in my last analysis, patients that received the neuromesh within phenotype number 2, their length of stay was longer than the patients that did not receive the neuromesh.